Hello, I, your instructor for this module, am Dr. P.R. Padma, Professor in the Department of Biochemistry, Biotechnology and Bioinformatics in Avinashalingam University for Women, Coimbatore. You have now chosen to study about the applications of transgenic technology or genetic engineering of animals in food technology. As you know, this topic is part of the food biotechnology paper. The introduction and the expression of transgenes in animals is continuing to be a daunting task, even though experimentally several success stories have been reported. As the animals are much more complicated systems than the plants, the commercialization of transgenic animals and their products is limited. This module covers the important developments in transgenic animals with reference to food technology and nutrition. After completing this module, you will have learnt about the approaches of genetic engineering in animals that have an influence in the field of food science and food technology, how genetic engineering has conferred the ability on animals to have improved nutritional value, increased growth rate and better quality and yield of milk and so on, and other applications including improved health status of livestock and poultry, reduced environmental impact and making bioreactors of animals that produce therapeutically important products. To give an overview of this module, the module deals with the advances made in the field of animal transgenesis related to food science and nutrition, as well as the attempts to develop new transgenic animals that are in the pipeline to be released in the near future. The module covers transgenic applications which we shall discuss broadly under the heads listed here. That is, enhanced nutritional value both in terms of quantity and quality, increased growth rate, improved health status of livestock as well as poultry, reduced environmental impact of animal excreta, improved feed efficiency and carcass composition, increased milk quantity and quality, secretion of pharmaceuticals and therapeutic proteins in egg white, and improved reproductive performance and fecundity. Some of these applications have become successful and are awaiting clearance for use, while most of them are still in experimental stage. Unlike plant transgenesis, the insertion and stable expression of specific genes in the animal system is highly complex. It is also time consuming and unpredictable. This is, at least in part, due to the complexity of the animal system. So far, only one transgenic animal, namely the aquaactive salmon, has reached the stage of regulatory scrutiny ready for clearance for marketing. This is in sharp contrast to the number of transgenic plants that have already been cleared for cultivation and are in use in several countries, including our own India. However, the potential applications of transgenic animals and their performance at the experimental stage have attracted the attention of scientists practicing transgenesis in animals. We will now take up the major transgenic attempts in animals that are related to the field of food science and nutrition. Let us start with the enhancement of nutritional value of animal products using genetic engineering approaches. As you must be already aware, animal food products have important nutritional attributes like the quantity, quality and nutritional composition. Genetic engineering techniques or transgenesis has helped in improving these traits in several ways. First, we shall consider the transgenesis approaches to increase the content of lean meat in livestock animals like cattle and pigs. A significant increase in the lean meat content of livestock animals has been achieved by the transfer of genes coding for the production of growth modulators such as growth hormone and insulin-like growth factor. Such a transfer results in increased quantity of lean meat from lesser number of animals than the conventionally bred animals. Different strategies have been developed to achieve this effect. These involve the genes that can mediate the formation of muscle tissue, improve the proportion of lean meat to fat, and result in a desired fatty acid profile in the tissue. Let us consider these one after the other. Starting with the transfer of genes that mediate the formation of muscle tissue. Insertion of genes that induce the formation of more muscle tissue in the animals has been achieved in pigs and cattle. Apart from such direct transfer of genes, another strategy that has resulted in a similar effect has been to insert the regulatory DNA sequences that overexpress the genes involved in the formation of muscle mass in the animals. This results in improved muscle tissue formation, yielding higher amount of beef and other similar animal products like meat used for human consumption. Transgenesis for the improvement in the production of lean meat to fat ratio in whole foods is the second approach that we shall consider. One of the major problems associated with the regular consumption of beef or meat is the ill health inducing effects due to the presence of high amounts of saturated fats in the products. Transgenesis has helped address this problem by the introduction of genes that alter proteins, regulating lipid metabolism, thereby improving the percentage of lean meat to fat in whole foods from animals. Successful transgenic experiments have been reported using the genes for the hormone leptin or the enzyme fatty acid synthase, which effectively increase the ratio of muscle mass to fat content, making the meat much healthier to consume. Transgenesis has also aided in the development of livestock with modified lipid profiles. An approach to address the fat content of animal meat or beef is to introduce the genes that influence this lipid profile as well as lipid metabolism, causing the synthesis of what we call heart healthy fatty acids. This approach results in the reversal of a fat rich protein poor diet to a protein rich fat poor diet, which is much desired from the health point of view. All of these potential transgenic interventions could result in more nutritious and healthful meat products used for food. The second class of transgenic animal experiments involve the conferment of increased growth rate. The introduction of growth hormone gene into a variety of animals has been attempted by researchers, resulting in increased growth potential as well as increased meat production. Such an introduction has been done in cattle, pigs and fish. The best success in this regard has been achieved with transgenic Atlantic salmon with growth hormone gene from fish. This transgenesis has resulted in a reduction of the growth time of the salmon by half. The implications of this observation are enormous, as this would mean rearing more salmon within less geographical area in lesser time. In other words, a tremendous increase in the yield of the salmon. In addition to this, there is a significant increase in the food conservation rate of these animals. That is, the achievement of enhanced growth will lessen food consumption, which has a profound influence on the environmental impact of such food-feed ratio. Let us now take up improved health status of livestock and poultry using gene transfer. When animal tissues are consumed as food, as beef, meat or chicken for instance, or are treated as sources of food as in the case of milk, it is imperative that we ensure that the animal is free of diseases. One of the major problems that the livestock rearing farmers have to tackle continuously is the pathogenic attack on the animals. Foodborne diseases are a serious concern all over the world as they adversely affect the morbidity and mortality of the population. In many cases, to counter the microbial attack, huge doses of antibiotics are administered to the animals, which, while getting rid of the microorganisms, can leave the residues of these drugs in the tissues or the milk. Thus, it becomes very essential that we ensure that the animals are free of both pathogens and of drugs like antibiotics. Transgenesis can help us in this regard, ensuring animal welfare by producing healthier animals. Transferring the genes to confer resistance to these diseases has been able to reduce the frequency of veterinary interventions like drug administration to animals, as well as reduce the supplementation of the animals with dietary and metabolic supplementary material. The transgenic animal is conferred with better resistance to diseases, which is a desired trait, especially in food technology and nutrition. The approaches in this regard include the development of poultry and livestock that are resistant to pathogenic organisms like Escherichia coli, Campylobacter, Clostridium species and Streptococcus. Genetic engineering approaches have also resulted in eliminating the susceptibility of the animals to diseases and the threat that they pose to the human health. The best examples in this regard are the diseases like the bovine spongiform encephalopathy or mad cow disease, or as it is called, or mastitis as it is also called, which is nothing but the inflammation of the mammary gland, reducing both the quality and the quantity of milk produced. Now, let us see how transgenesis has reduced the environmental impact of animal byproducts, especially that of excreta. Managing the animal excreta is a serious issue in animal husbandry and agricultural practices. Several uses have been devised for the excreta, like the use as manure or the substrate for vermicomposting, or as a source of natural gas or methane, which is popularly called gobar gas. In spite of such uses, the issues associated with the handling of huge volumes of excreta remains unravelled completely. Especially important in this regard is the excretion of very high amounts of elements like phosphorus, which can have serious ill effects on the health of the animal handlers as well as impact the environment adversely. This issue has been dealt with using transgenic approaches, wherein the animal is bestowed with the ability to produce more protein-rich food products like milk and meat. This not only results in enhanced growth of the animal, but also drastically reduces the amount of phosphorus that is excreted. Thus, the transgenic attempts in this direction are expected to reduce the amount of manure by increasing the ability of converting the food consumed by the animal to building blocks or products, decrease the direct competition between humans and animals, save water by cutting down the consumption of water both for the animal growth and maintenance of hygiene in the animal housing facility, as well as diminishing the land footprint that is needed for the livestock facilities, thus also decreasing the damage to the environment. Even though several livestock animals and poultry have been treated in this regard, the best success has been achieved with the pigs. The introduction of gut enzymes like phytase or xylanase or both phytate and xylanase in animals like pigs and poultry increases the bioavailability of phytic acid from sources like corn or soybean products. The expression of phytase in the salivary glands was achieved in pigs as early as 7 days of age in the piglets. Such an expression in the saliva resulted in a very efficient phytate phosphorus utilization as well as decreased the phosphorus excretion by these animals by more than 75%. Additionally, these animals did not need phosphorus supplementation in the diet to achieve normal growth. In this direction, a transgenic pig variety that is rightly named the Enviro pig has been developed, which not only produces more pork but also excretes a significantly low amount of phosphorus into the environment. Thus, transgenic approaches have also concentrated on the sustenance of our environment. Improved feed efficiency and carcass composition has also been achieved by genetic engineering of animals, as we shall see now. Understanding the role of genes in influencing the growth of animals has paved the way to several transgenic approaches to increase the feed efficiency as well as the product composition of the animals. Better growth characteristics have been achieved by the transfer of genes of growth hormone, growth factor receptors as well as growth modulators. Transgenic livestock, including cattle, sheep, pig, poultry and fish, have been developed using this approach. This has resulted in improved feed efficiency as well as increased growth characteristics. Several genes have been identified in the animal system that affect the yield and quality of the meat that is produced. Notably, a gene called acid meat gene or RN gene, which is short for rendement napole gene, has been identified as the contributing factor for lower yield of processed meat from pigs. This is because the RN gene product influences the pH after the sacrifice of the animal, resulting in hardness of the meat. The transgenic approach adopted for tackling this problem is to create knockout or KO organisms in which the RN gene has been silenced. This results in an altered post-mortem pH which produces tender meat. Other genes that have been used to achieve improved growth characteristics and carcass quality include the genes that code for ryanodine receptor, growth hormone releasing factor, high affinity insulin-like growth factor binding proteins and myostatin, which is growth or differentiation factor 8 or GDF8. The myostatin gene has been considered as an exceptionally potential locus for knocking out in the meat-producing species. The loss of the myostatin protein results in an increase in lean muscle mass, which is very desirable carcass property. One of the most promising transgenic strategies in animals is the increase in milk quantity and quality. Milk is an essential animal product that can be obtained by non-invasive methods every day. Enhancing the milk yield and its components has been a priority in transgenic efforts. The focus of transgenic approaches in improving milk has concentrated on increasing the amount of milk secreted, enhancing the nutritional value of the milk, and secreting pharmaceutical or nutraceutical compounds in the milk. These attempts can be considered under several subcategories. The first of them is increasing the amount of milk secreted. An increase in the amount of milk secreted by an animal will benefit both its offspring as well as the humans who are consuming the milk. Transgenic cattle, sheep, and goats are used for this purpose. Even small increase in the volume of milk, for example 2 to 4 liters per day, is known to have a profound effect on the weaning weight of the young ones and improved growth of the offspring, resulting in a shortening of the duration of utilizing them for food purposes. This also has been observed to improve the survival of the offspring of the transgenic experimental animals. The second strategy is by enhancing the nutritional value of the milk. Milk, as we all know, is a good source of proteins, fat, and the milk sugar lactose. Enhancing the amount of all these components have been achieved by experimental transgenesis. The aim of these transgenic approaches is to get the milk amenable to manufacturing processes using the milk as a source of its components. Successful transgenesis has been carried out to increase the amount of specific caseins in milk. Such casein-rich milk is projected to be very useful in the manufacture of high-quality yogurt and cheese. Another approach is to increase specific types of casein like the beta-casein and the kappa-casein in bovine milk. Increased amount of glycosylated kappa-casein increases the solubility of casein in milk, which decreases the whey expulsion during coagulation for the manufacture of cheese as well as thick curds. It is also expected to result in better tasting low-fat cheese. Increasing the quantity and quality of milk alone has not been the only aim of the scientists. Secreting pharmaceutical or nutraceutical compounds in milk as well as eggs has also been achieved against diseases and disorders using gene transfer methods. A sizable proportion of the population suffer an inherent defect of the enzyme that cleaves the milk sugar lactose into its component sugars glucose and galactose. This results in a condition called lactose intolerance. Individuals who are lactose intolerant have problems digesting milk and need to take lactose cleaving enzymes as drugs throughout their lives. A solution to this problem has been attempted using transgenesis. The intestinal lactase enzyme has been transferred and expressed in the mammary gland of the bovines. This resulted in the reduction of lactose in the milk produced by this transgenic animal, benefiting lactose intolerant subjects. Other therapeutic products that have been secreted into milk by genetic engineering include insulin-like growth factor, namely IGF-1, epidermal growth factor or EGF, transforming growth factor beta or TGF-beta, and lactoferrin, which improves the growth, health status and the survival rate of the offspring as well as the humans who consume the milk. Several antimicrobial compounds and peptides like alpha-lactalbumin, lysozyme and lysostaphin have been conferring resistance against a wide spectrum of bacterial diseases, resulting in better animal and human health. Apart from these, milk from transgenic animals has also served as source of antibiotics against several diseases like mastitis in cattle, sheep and goats, and mastitis metritis A galactea in pigs, and antibodies that prevent these many domestic animal and human diseases. Using transgenesis, antibiotics and therapeutic proteins like anti-snake venom have been secreted into the egg white by genetic engineering of the poultry. The secretion of such therapeutics into egg white has also been achieved by non-transgenic approaches and have had limited success. In recent years, attempts to secrete the therapeutic proteins into egg white using transgenesis attention of scientists. Another important area where the transfer of genes into animals has had exciting possibilities is the improvement in the reproductive performance of animals. Sustaining livestock and poultry with superior qualities depends on the efficiency with which they reproduce. In traditional breeding practices, some of the poultry varieties reach reproductive senescence at an earlier time point. A similar trend has been noticed in some farm animals also. This has been overcome by genetic engineering techniques. Genes have been targeted for transgenesis of chicken as well as other farm animals to postpone reproductive senescence. Additionally, the introduction of estrogen receptor genes and fecundity genes has improved the reproductive performance of transgenic animals. Thus, several transgenesis approaches have been developed in the animal systems, resulting in the expression of desired traits in the resulting animals, as we have seen in this module. However, the commercial production of such transgenic animals has not seen the light of the day. Only one or two of these have even reached the stage awaiting official clearance for large-scale production and use. Now let us see why this is so. As already mentioned earlier, animal transgenesis has seen only limited success. This is due to several reasons, the major ones of which include the ones that we shall see now. The animal systems are much more complicated than the plant systems, which makes the introduction and expression of new genes very difficult. Totipotency is a very rare phenomenon in the animals, observable only in certain specific cells in the very early embryonic stage. In plants, all the cells express totipotency throughout the life, making them easily amenable to transgenic manipulation and expression, which is not the case with animals. The animal systems, especially the terrestrial animals, are governed by very strict ethical guidelines, limiting the possibilities of experimentation and implementation to a large extent. Additionally, there are several organizations like the SPCA, which stands for Society for the Prevention of Cruelty to Animals, that oppose the genetic manipulation and experimentation in animals, which also influence the number of attempts to make transgenic animals. In spite of such limitations, scientists all over the world have attempted transgenesis in animal systems at the experimental level, and the applications of such attempts have been dealt with in this module, and the success stories that are in the pipeline are also understood. To conclude, animal transgenesis for food and feed applications is largely still in the experimental stage. Though huge successes are reported on the development of transgenic animals in several forms, the commercialization of these has not become a reality. The only application that is close to being cleared for use is the Atlantic salmon with high growth potential. The major issues to be addressed and circumvented are the safety aspects and the ethical concerns that loom large over the transgenic initiatives in animals. These aspects will be dealt with in the next module. Thank you.